Hi guys, this is my mini lecture on energy, but the specific section is on diffusion and osmosis, active transport and passive transport. So here we go. So I'm telling you that it takes energy to move substances in and out of the cell. And it's very important what gets in and out of a cell because it could come in and burst a cell if too much comes in or too much comes out, and it could shrink a cell. So very important as to what can get in and what cannot. So we've already looked at the phospholipid. It makes up the membrane. This is just a short review. You have your phospholipid bilayer, and this section with the balls is going to get along with water that's on the outside of the cell, and the balls on the inside are going to get along with water on the inside of the cell, because that phospholipid head is the one that gets along with this aqueous solution. Okay, and here's some proteins that go across that membrane. So that's what makes up the membrane. And we also looked at more specifically at how we look at it as a ball, and the two legs, the stylized model like this, where the fatty acids are shown as the legs, and the head here really is that phosphate that has a charge to it and making it polar. So the function of this cell is to, as I said, it's a boundary. It's a boundary that's not going to let some substances in and not let some substances out. And that is extremely important for the health of the particular cell. So the stop sign there is just showing you not allowed in or yes allowed in. There's a really interesting story of a sad story that happened many years ago now. There was a radio contest where, as a lark, as a hoot, they said they had a contest and who could drink more water on the radio station. And the person who drank most of the little tiny water bottles would win a Wii for the computer. And so these contestants came in, and this is really funny, and we're going to drink water, and whoever drinks more water is going to win the Wii. So it was called Hold Your Wii for a Wii. The contestants that came in was one big guy, and he had greater mass than this other younger gal who was a young mother and wanted the Wii for her kids. And she went through the contest. She came in second. She couldn't drink that much water, went home, lay down because she was feeling terrible from drinking so much water, and died. And I bring up that story because that is a perfect example of too much water diluting everything. Too much water coming into these cells, too much water diluting salts, and this individual didn't survive that. And it was a big tragedy. And it was actually settled in court for millions of dollars later on. So what's the take-home message? The take-home message there is that concentrations are important to maintain, and if you're going to maintain a concentration in an organism, it's going to be very important that that cell regulate what gets in and what gets out. Again, the membrane with the bilayer that I'm showing you, and it's going to take energy to do this, right? Sometimes it doesn't. If you have molecules that are coming through, sometimes they can't come through the membrane itself, but what they will be able to do is use a protein like a channel. So we call it a channel protein, and they'll be able to use it like a roadway, like a river, to be able to come in. So let's look at the different types. How does it work? The first way that it could potentially work is passive, which means it doesn't need energy. So all you would need is some movement of molecules from the outside to the inside of the cell, let's say. It's going to be passively moving, and you're not needing any kind of energy. And I'll go into more detail of that in a little bit. The examples of that would be called simple diffusion. It's also used for osmosis. So simple diffusion of water is osmosis. It could be active. If it's active transport, it is going to use energy. It usually requires ATP. So my picture of someone desperately biking up a hill is going to be active transport. So active transport is meaning that usually that what we're transporting already is in a high concentration, let's say inside the cell. It's already very concentrated inside the cell, and yet you need to bring more in. That's going to be kind of against the concentration gradient, is what we call it. It's difficult when there's already a lot of that substance in there to bring more in, and that's going to take energy to do that. So that's called active transport. Vesicular transport is when what you need to bring inside the cell is so big that not even a channel protein will allow that. So you have to essentially blast through the wall, through that cell membrane. You're just going to have to sort of destroy a piece of the membrane and just come on in. Things like white blood cells will do that with large molecules. They have to move, and they'll just push them outside of the cell. And I'll show you some examples of that. So vesicular transport then is, it can't come through the membrane. It can't come through a channel. It has to pretty much blast through the membrane and push away some of those phospholipids. So what are the requirements for the simplest of these, which is simple diffusion? Simple diffusion has to have a concentration gradient, meaning there has to be more cells in one end and fewer on the other for those molecules to just, as they bump along, move towards where there are fewer of them. So that is a concentration gradient. More on one end, fewer in the other, you move to where there's fewer. And that can happen because of random molecular motion. So you all know that in every day of your life, right? If I were to walk, if I were to come into a classroom where we're all meeting in the classroom and I'm full of perfume, then the people in the front row are going to smell a little bit of it, and little by little that's going to move towards the end of the classroom, because one, there's a concentration gradient, there's lots over here and less over there, and two, there's random molecular motion. Those molecules are bumping into each other. They're always moving, and they will just slowly move until they've pretty much reached an equilibrium. So those are the requirements for simple diffusion. We often think of it in terms of a membrane because we're thinking of a cell. That this is area 1, very concentrated. Area 2, not so much. So these little balls will bump into each other and move from area 1 towards area 2 until they are in equilibrium. We call these solutes. So random molecular motion eventually results in something like this, where it is evenly distributed. Does that mean they stop moving? No. They continue to bump into each other, but they're distributed pretty evenly. Now imagine if you had to do it backwards, if you had to get all these balls more into that way, into where there was already a higher concentration. That's when you would need energy. But more on that later. So when we talk about osmosis, it's exactly the same thing, but relating to water. So osmosis is the diffusion of water. And in more detail, osmosis is the diffusion of water from where it's highly concentrated, where there's a lot of water, to where there is less water, lower concentration of water. And it will always do that. Now, we add a membrane. And we add a membrane to the story because we're thinking of cells, right? And cells will have a membrane. So here, if we did it in this little U glass, and we put a membrane right in the middle, and we put water in both sides, and water was able to move from one side to the other. But this sugar that we put in it, glucose molecule, is actually too big a molecule. You made it, you built it already, so you know it's a pretty big molecule. And it wouldn't be able to cross this membrane. It's stuck there. This is a filter, and the sugar can't get through, but water is very tiny, and water will be able to flow through. So we say that solution B has more solute, right? But really, solution A has more water, correct? It has more water, less solute, and solution B has less water, more solute. So we expect, if we can't bring the solute over to this side, and we need to equilibrate these solutions, what is the only option available? The only option available is for water to move. And water is going to move from A to B, where there was more water to where there was less water comparatively. And this is what's going to happen. When we talk about concentration of solutions, it's going to be really important, right, in a body, because if we put a cell in a place where there's less water, water is going to move there. If we put a cell in a place where there is too much water, water is going to move in. So it's very important that we bathe our cells in a solution that is just right. So this terminology here has to do with solutions that you would put your cells in. You know that the perfect solution for your cells is that of the salinity of your tears. And that's perfect. Just enough salt. Ocean, too salty. Tap water, not salty enough. But that tear is just about the right solution for it. So we have terms for too concentrated, not concentrated enough, and just right. Hypertonic is a solution with a higher solute concentration than a cell. So usually salt, too salty. Solution hypertonic, too much salt. And if you were to put a cell in that, water would leave the cell, going towards a location where there was less water. Opposing that would be hypo, less tonic, lower solute than a cell. And then water would move into the cell. This is what happened to the lady who drank too much water. All of a sudden, she had this condition of hypotonicity because she drank and drank and drank water, and she diluted all her salts and all her sodium and chloride, and her cells became hypotonic, and they started to rupture. Isotonic would be just right. Your tears, same concentration, no net movement. That's what makes our cells happy. Okay, remember.